Hi everybody, I'm back again. Hi, it's Lindy Lumsden here. I'm the science leader at ARI and I'm filling in for Fern Hames today, who's off doing other things. So it's great that you can all join us. We're going to have two fantastic seminars today on being able to use genetics in conservation management. So they're going to be really, really fascinating. But first I'd like to acknowledge the traditional owners of the lands on which we're all variously meeting. So for me, that's the Wurundjeri people of the Kulin Nation, and acknowledge their elders past, present and emerging. So our seminars today are by Sasha Pavlova and Zeb Tonkin, and we'll hand over to Sasha first, who's going to talk about genetic management of wildlife in a changing world with a particular focus on helmeted honeyeaters, and it's going to be a fascinating seminar. So over to you, Sasha, it's going to be great.
Hello everyone, my name is Alexandra or Sasha Pavlova. I'm a research fellow at Monash University. I would like to start by acknowledging traditional owners of the land on which we all meet today and pay my respect to the elders past, present and emerging. I also thank Indy and Lindy for inviting me to speak. In this talk, I'm going to tell you about our work on genetic management of a Victorian bird emblem, the helmeted honeyeater. I will start by outlining some basic concepts behind genetic management. Then I'll show you how we applied it to save the helmeted honeyeater. Next, I will explain how genomics can help in designing management of threatened species and how to conduct genetic management if genetic data are not available. This work was possible thanks to sustained commitment, energy and goodwill of many stakeholders, including DELP, Zoos Victoria, the Helmeted Honeyeater Recovery Team and community groups, notably the Friends of Helmeted Honeyeater. On this picture, you could see three Helmeted Honeyeater heroes. Bruce Quinn, who has dedicated 30 years of his life to saving this bird, Alan Clayton, the president of the Friends of Helmeted Honeyeater, and Karina Cartwright, who breeds honeyeaters in captivity. The work I'm presenting here today was done in close collaboration with Professor Sunnucks, one of the world leaders in wildlife genetic management, and these brilliant researchers, Catherine Harrison, Diana Rabeda-Roos, and Michael McGrath. Genetic diversity is one of three main pillars of biodiversity, the other two being species and ecosystem diversity. Genetic diversity underlies diversity of phenotypes, such as external appearance, behavior, food preferences, which allows species to explore various niches. It also underlines population health and fitness, or ability to persist and grow by providing a diversity of immune responses, behaviors, and breeding strategies. Genetic diversity is also essential for adaptation to new environments, as some phenotypes will be less fit than others. By destroying and degrading habitats, humans make populations small and isolated. This leads to chance loss of genetic variation, which in turn results in population differentiation and low capacity to adapt to new threats. Small populations face additional genetic problems. As individuals become more similar, reproduction between close relatives, or inbreeding, becomes more frequent. Inbreeding for many generations could lead to loss of fitness. Inbred individuals have fewer offspring, and offspring could have weaker health and die earlier. This is because harmful genetic variation gets expressed in inbred individuals. Further loss of variation could result in further population decline and make it vulnerable to extinction. In this cartoon example, there was nothing wrong with the yellow birdie. It was just unlucky, and bad luck has more impact in small populations. Most naturally outbred populations experience loss of fitness due to inbreeding, as animals in these three examples. This means that even if threats are removed and habitat is restored, the population will struggle to recover. Luckily, there is a natural solution to genetic issues of small populations, and that is gene flow with one or more populations. Ideally, we would be aiming to restore natural gene flow from a genetically diverse population with which a small population was previously connected. If such gene flow results in higher fitness of admixed individuals, it is called genetic rescue. Genetic rescue increases population growth and resilience to future changes. Augmented gene flow could be achieved through habitat reconnection or direct animal translocations. Such gene flow increased fitness in great majority of reviewed empirical cases. Moreover, positive fitness effects were stronger in stressful compared to benign environments. There are published guidelines on how best to conduct assisted gene flow. It is considered safe to connect populations that diverged recently, are similarly adapted, and have been in gene flow in the past. Connecting populations that diverged long ago, are differently adapted, and have different number of chromosomes may be more risky. In the worst cases, this can result in lower fitness of admixed offspring. But in an increasing number of cases, risky rescue may be the only available option, because doing nothing is more risky. The good news is that negative fitness effects of gene flow can be transient, and apparently appropriately monitored, they could be adjusted through change in management. So what is genetic management? It starts with genetic analysis, which estimate how different populations are and how long ago they separated. Post and present gene flow, genetic diversity and inbreeding, and presence of local adaptation across the species range. Next, a risk assessment is conducted, where the risk of lost fitness due to gene flow is weighted against the risk of lost fitness due to inbreeding. Finally, management recommendations are made on which populations require gene flow and where to source the migrants from. Helmeted honeyeater is one of the best examples of how these principles can be successfully applied. Helmeted honeyeater is the subspecies of the common yellow-tufted honeyeater, distinguishable from other subspecies by presence of erect feathers on its head, so-called helmet. Helmeted honeyeater is critically endangered. Only 250 wild birds remain in a single remnant population located in uniquely swampy habitat at Yellowbow. The population is being intensively managed with efforts including habitat restoration, captive breeding program, and releases. Despite these efforts, numbers remain very low. Populations started growing after supplementary feeding was scaled up. This is very labor intensive and hard to sustain, so ideally we would not have to do it. Indeed, we may not have to if we had genetically healthier birds. Over the same time period, genetic diversity declined dramatically in both wild and captive populations, suggesting that population ability to adapt to novel threats becomes weaker with time. In addition, inbreeding in the wild population increased to a degree that is usually harmful for fitness. Thanks to tremendous efforts of Dell, and in particular Bruce Quinn, 30 years of field fitness data for the helmeted honeyeaters has been collected. We used this unique data set to test for fitness effect of inbreeding. These data clearly showed that fitness of helmeted honeyeaters declined with increased inbreeding. More inbred birds died much sooner and have far fewer offspring in their lifetime. When these inbred birds produced 27 fledglings, the most inbred birds produced only 2.5. Inbred individuals were less likely to breed at all. We also asked whether wild birds avoid inbreeding and whether management can help them to do it better. Field data shows that choosing an unrelated mate can improve fitness of a helmeted Honeyeater pair. But do the birds choose the best mates? Unfortunately, it doesn't seem so. Observed level of inbreeding, shown here as a green bar, was not different from the random mating, shown as an orange bar, but was significantly worse than what would actually be achievable if birds did avoid mating with relatives, a yellow bar. So can the birds be helped? For example, would genetic quality of pair improve if we could remove one of the members of the most related pair and let the other one repair with another individual? We used field breeding data across many seasons and simulations to test this. Unfortunately, the answer is no, because social organization gets on the way. Helmeted Honeyeaters have social neighborhoods arranged along creeks, as shown on this map. Most breeding pairs, shown here as green bar, were actually detrimental for population health because their offspring will increase genetic problems in the population. Best achievable combinations of pairs within neighborhoods, shown as a yellow bar here, makes pairs only slightly beneficial to population health. But high red bar shows that management, separating most inbred birds and allowing the remaining bird to repair within the neighborhood, would not help remove genetic problems. This is because territorial defense may prevent dispersal of young birds from different neighborhoods and force local birds to breed with their relatives. This means that genetic rescue could be very helpful. So we set to conduct the risk assessment following recommendations of Frankham and colleagues, targeting 5 questions. Is the population inbred? Are there other populations that could be used for crossing? Will crossing lead to loss of fitness or uniqueness? Will increasing gene flow lead to worthwhile benefit? And how gene flow could be optimized? We started by looking for an appropriate source of gene flow. Given that helmeted Honeyeater, color-coded yellow here, has only one population of its kind, gene flow has to come from another subspecies. The closest appears to be Gippslandicus, color-coded red here. Gippslandicus and helmeted Honeyeater diverged around 50,000 years ago, but were historically connected by low levels of gene flow. They currently occupy slightly different habitats and differ in mobility. Successful hybridization was observed between the 2 subspecies in the reintroduction site. Still, this type of rescue may be considered risky. Thus, monitoring fitness during rescue will be very important, so management could be adjusted accordingly. But the right question to ask is still, will the benefit of genetic rescue outweigh any possible harm? Population viability models that include inbreeding depression could help us make the right decision. We simulated several management scenarios with and without genetic rescue. These showed that population will likely go extinct within few generations without supplementation. Even with reasonable level of supplementation with pure helmeted Honeyeaters bred in captivity, extinction is still possible. Yet all scenarios that involve gene flow from Gippslandicus result in population persistence. This supports gene flow as likely beneficial for population health. Genetic rescue trials in captive breeding program were co-designed in collaboration with Zoos Victoria. They started in 2017 and are still ongoing. Over the 5 years, 116 pure helmeted Honeyeater pairs were set up alongside 44 hybrid pairs. Full analysis of the outcomes, accounting for various biases, are still ongoing. but a preliminary look at the data shows that hybrid pairs outperformed the pure helmeted honeyeater pairs on many fitness indicators. Hybrid pairs were more likely to lay eggs, attempted larger number of clutches, and produced more fledglings per pair. The improvement in fitness of hybrid pairs compared to the pure pairs may appear small. However, empirical and experimental data suggest that stronger fitness gains could be expected for genetically diverse individuals in stressful environment of the wild compared to benign captive environment where most stresses are removed. Genetically diverse individuals born in captivity were released into the last remaining population of helmeted honeyeaters, and many successfully bred. By February 2021, 10 genetically diverse birds were produced. And last winter, a second helmeted honeyeater population was successfully established through wild-to-wild translocations and captive releases that included genetically diverse individuals. The next step is to monitor the fate of genes unique to helmeted honeyeater and manage gene flow so that genetic variation beneficial to helmeted honeyeaters is retained. High-quality genome and linkage map are genomic resources that can inform analysis of genomic data, and we produce these resources for helmeted honeyeater in collaboration with DNA Zoo and others. Diana Rabeda-Ruz made a short video explaining how these resources will help in genetic rescue. We will use genome sequences for a small sample of individuals of all subspecies to infer how populations have changed over time and identify useful and potentially harmful variation. Next, we will use field fitness data to validate common assumptions about which variation is harmful or not. Together, these data will help answering whether current level of gene flow are appropriate for fixing and breeding while retaining unique genomic features and promoting adaptation to changing climate. Here are a couple of examples of genomic analysis informing management. First, total length of inbred genome segments calculated for birds across all subspecies showed that helmeted honeyeater is the most inbred of all subspecies, and inbreeding increased with time. We can reverse this with genetic rescue, validate it with fitness data, and monitor the change through this metric. Another example is reconstruction of how population changed over time. This analysis done for a single helmeted honeyeater genome shows decline of helmeted honeyeater population during ice ages. Similar analysis for other subspecies will help estimate when subspecies split and when they stopped exchanging genes. In summary, we use genetic risk assessment to design genetic management for the helmeted honeyeater, and now we can answer our 5 questions. Is the population inbred? Yes. This answer should be good enough to take action. Demonstrating strong inbreeding depression with a bonus, which should not be necessary for every population. Are there other populations that could be used for crossing? Yes, Gippslandicus subspecies is most closely related of the remaining population and is genetically more diverse. Will crossing lead to loss of fitness or uniqueness? This is unlikely because there were no natural hybrids between helmeted honeyeater and Gippslandicus, and recent gene flow estimated between them. This means that local adaptation occurred in the presence of gene flow and thus could be retained if gene flow is restored. Will increasing gene flow lead to a worthwhile benefit? The answer was yes, because population viability analysis suggested gene flow will alleviate inbreeding depression. Finally, how to optimize gene flow. We will monitor fitness outcomes using field estimates and genomics and adjust management accordingly. The same principles can be applied to other threatened species. For example, we're also working with wildlife managers on co-design of genetic rescue of other species and developing genomic resources for future monitoring. In the next talk, Zeb Tonkin will tell you how genetic management was applied while restoring extinct Macquarie perch population. You may ask, okay, helmeted honeyeater has a lot of data, but how about species which don't have such rich genomic resources? Actually, the same principles can be applied even when genetic data are limited. Victorian government has invested in the Victorian Genetic Risk Index, which uses available information to derive management recommendations for over 1,000 Victorian species. This index is now mainstream in decision making. For example, it was used in recent bushfire recovery efforts and sanctuary planning. Finally, I would like to flag this book specifically designed for wildlife managers as a step-by-step guide on genetic management. Please don't hesitate to talk to us if you need to consider or implement genetic management. Genetics and genomics are not that difficult and not that expensive, but it can greatly facilitate population recovery. We are currently working on the ways to streamline and scale it for broader use in conservation. I would like to thank many collaborators who have contributed to the work I presented here today, especially Paul Sunnucks, Catherine Harrison, and Diana Robledo-Views from Monash University, Michael McGrath and Dan Harley from Zoos Victoria, and Bruce Quinn from DELP. I also thank funding agencies and many stakeholders, including DELP, Zoos Victoria, and others, and I thank them for a sustained commitment to biodiversity conservation. With this, I would like to answer your questions.
Fantastic. I'm not sure if I'm on yet, Andy. Fantastic, Sasha. That's wonderful presentation. Thank you so much. Yeah, fantastic presentation. I really liked the background in terms of, explaining some of the concepts around, genetic management, because not everybody's, that familiar, but I thought you presented that beautifully. So thank you, and just great to see the how helmeted honeyeaters are going and with the genetic rescue interventions. Just while we're getting questions coming through, I'll just ask you one quick question. You mentioned about the uniqueness, and I think uniqueness is something that I know in the past, people have often been concerned about interbreeding, losing the uniqueness, but you obviously, think that that's not a big issue. But I was curious, is it that if there's, hybrids crossing, would they then cross back more with pure helmeted honeyeaters, or would it keep on being crossing of hybrids, and so keeping on introducing the Gippslandicus genes into the mix?
That is an excellent question. We are trying to manage gene flow in such a way that only a limited number of Gippslandicus individuals and migrants go into the helmeted honeyeater population. So it is more likely that back crosses will be formed rather than hybrids, second generation hybrids. So we think that the helmet will persist in helmeted honeyeaters, as well as other adaptive variation. So it could be managed, and that's why we would like to measure, keep monitoring the population and see what happens.
Fantastic, thank you. So, Andy, over to you. Are there more questions at this stage, or are people still thinking up questions? Often it takes a while to think up fantastic questions, and everybody's been so absorbed in the presentation. But over to you, Andy, with some questions.
No worries. Thanks, Lindy. We've got one from Glenn. He's asking, Sasha, you partly explained a little bit of this question, but he's asking around whether the melon tony, the West Great Divide yellow-tuft honeyeaters, were they not a good option for crossing compared to Gippslandicus? Was that direct comparison between the two subspecies done?
It's also a good question. So apparently Meltoni and the melanops, which is another northern subspecies, they are morphologically slightly different from both Gippslandicus and helmeted honeyeater. They're slightly smaller and they're more mobile, but they're genetically very similar to each other. So still, they would be not the first priority. And in locations where helmeted honeyeater occur, Meltoni never were seen hybridizing when they show up in that location. They never have been seen to hybridize with the helmeted honeyeaters before. So they would be not the first priority, I would say, but there's still possibility if we lose Gippslandicus for some reason.
Really good answer. Thanks, Sasha. I've got one around the cost of doing genetic research. How expensive is it to get a sample to carry out a genetic readout for a honeyeater? So I think this is a question about per bird cost of doing a genetic assessment.
Also a good question. So the costs are always declining, the cost of sequencing becoming more and more affordable with time. The cost per sample depends on what exactly you're trying to answer and what kind of data you're trying to achieve, trying to obtain from that sample. For example, genome-wide subsample of the genome will be cheaper. It would cost you maybe $50 per individual, whereas whole genome sequencing may cost you maybe $200, $300 per individuals, whereas the whole genome assembly would be probably more expensive. And first genome for the species would probably cost around $1,000 or so.
Thank you so much. I'm glad you had those figures at the top of your head. I've got a question now around the social license of genetic rescue. In this example, you've talked about crossing subspecies, but this person's wondering about the crossing of different species in order to get a bit of genetic rescue. Do you think there's a license to do that?
It's a good question. And I think it depends on personal perception. We as a community of people should have a dialogue about this. In my view, it's better to have this animal than not to have, even if it's a hybrid of some sort, rather than not have it at all. So in my view, hybrids are as good as the species, because if they're there, they're doing what they're supposed to be doing, birds catching insects and contributing to the ecosystem. This is all great. So in my view, I think we should be having more of the gene flow experiments, because we were the ones who deprived many species of genetic diversity in the first place.
Really good answer. Thank you. I'm actually going to continue on the line of thought that you just gave. You mentioned that it's good to keep the species, but one of the great things about helmet honey is that little tuft that you get. Are you monitoring the changes in that in the hybrids and whether or not it still looks like a helmeted honeyeater?
Yeah, there's some people who are collecting this data, and I've only heard of that data, so I haven't actually seen or read anything written up. But I think they still have, the hybrids are still having helmets. But don't quote me on that. I need to ask experts.
No, perfect. Thank you so much, Sasha. Thanks for answering all the questions, and thank you for the audience for asking them. I'm going to throw to Lindy to introduce our next presenter.
Okay, fantastic. Thanks, Sasha. Now we'll move over to Zeb Tonkin from ARI, and Zeb's going to be talking about incorporating genetic management into threatened species recovery, and particularly the case of Macquarie perch. And so this is going to be a fantastic extension of the sort of work that Sasha was talking about, and Sasha's a co-author as well. So over to you, Zeb.
Thanks, Lindy, and thanks, Sasha. Like Sasha, I'd also like to acknowledge the traditional owners and pay my respects to elders past, present and future from the lands that we're all dialing in from, and also where this work was undertaken. I'd also like to acknowledge my co-authors at this stage, those from Victorian Fisheries Authority, Monash University and DELP, and a whole bunch of others that I'll flag at the end of the talk, individuals and organisations that have all made massive contributions to the success of the project. First, I'd like to say that unlike Sasha, I'm not an evolutionary biologist, so save any questions on this front for her. But this talk is based on some threatened species recovery work that incorporated those pillars of genetic management that Sasha so fabulously introduced just then. So it provides a great example of how this work looks out there on the ground. So specifically, I'll be talking about our actions that aim to reintroduce the makings of a self-sustaining population of the nationally endangered Macquarie perch into the Ovens River up in northeast Victoria. I'll talk a bit about the species in the system where we completed the actions, the specific actions that we used, and finally a wrap up of the evaluation that we did. So I'll jump straight into the species. So Macquarie perch are a species of immense cultural, recreational and conservation significance. They're a medium-sized native freshwater fish, endemic to the Murray-Darling Basin and a few catchments in the east coast. They're a riverine species, but they can establish populations in lakes where they can, as long as they can access streams and rivers that they require to undertake their reproduction. They do this between late spring and early summer for several months. They're a long-lived species, up to 30 years of age for a fish that's around 40 centimetres long. And historically, they were a dominant species in those mid to upper reaches of the Murray-Darling Basin. And unfortunately, like many of our native species in the basin, we've seen major declines in both their abundance and also distribution. We've seen this by the mid-1900s and massive ones by the 1970s, to the point where they're now listed as nationally endangered and also in Victoria. We're now restricted to these 11, what we're calling populations in Victoria that historically used to be connected, but these 11 populations are highly fragmented and isolated, and as a result got this low genetic diversity. And where these populations exist, or where many of them persist, they're often in suboptimal habitat with no sort of connection to other source populations. So the future of many of those populations is in doubt. So what went wrong? I guess all the usual suspects. So we've got introduced species, particularly redfin perch, disease, habitat degradation, particularly some of that associated in the early years, associated with the gold rush, where we had desnagging, siltation, loss of riparian zones and subsequent water quality. We've seen a lot of overfishing and illegal fishing. And then the big one is river regulation and everything that goes along with that. So with all of this, these are all sort of past actions, I guess, past threatening processes, but we've now got this new threatening process where we've got this isolation and loss of genetic diversity that really does hamstring, I guess, the future recovery of the species. So what are we going to do about it? So, you know, it's been going on for a few decades now. So we're putting habitat back, in-stream habitat restoration and riparian repair. We're controlling exotic species. We've got fisheries regulations and enforcement in place now. And we've been quite responsive in terms of actions like translocation and fish rescues to episodic events. So when we see a big sediment event coming off the back of a bushfire, we try and shift individual fish out of those areas. There's also been a lot of restocking and translocations undertaken in the past, particularly in the 1980s and early 90s. But a lot of these actions were undertaken in small systems. They were only a single sort of stocking or translocation event. And there's been limited success from some of those early interventions. Moving forward, we're sort of recognising that there's a need for these longer term solutions that are focused on increasing population resilience. So we want to try and expand populations now, and also connectivity between such. We want to focus on some of our larger systems that are a lot more resilient to future predictions of climate change. We want to continue some of this education of key stakeholder groups, and also, as Sasha just mentioned, that genetic health, we need to start considering that. So if we hone in on Macquarie perch in terms of the genetic management, so Sasha led a study back in, I think this was published in 2017, where she had a look at the effective population size and genetic diversity of most of our remaining populations across southeastern Australia. And basically what we found is that most of them all had really low genetic diversity and effective population sizes. Small populations in particular may experience a loss of fitness due to inbreeding depression within the next few generations. And as such, most of those populations require genetic restoration to boost their genetic diversity and adaptive potential. So I guess with these genetic issues in most of our populations in terms of inbreeding, genetic load and loss of adaptive potential, the conclusion from that paper I just mentioned was that mixing populations was recommended, and these recommendations have fortunately been adopted into the National Recovery Plan for the species. So that leads us to this project. What did we do? So basically our aims were to re-establish a self-sustaining population of Macquarie perch into the Ovens River, where they've basically been lost. We wanted to do this by translocating and stocking fish back into the river, basically to establish a broad age and size structure, just like we see out there with remnant populations. And we also want to consider this genetic diversity. We want to maintain that. Along the way in doing so, we wanted to raise community awareness and support and participation of the project. And we wanted to really value add and expand on earlier threatened species and river health programs, which sort of leads me to sort of highlight why the ovens. Why did we pick this system to have a crack at this? So initially, obviously, Macquarie perch are now locally extinct in the system. They have been since the 1970s, maybe early 80s. But there's been a whole lot of work done over the last 3 or 4 decades and we've never turned the species up. It's a largely unregulated river, so the flow regimes in there is actually pretty good. It hasn't got these whopping great dams and issues with cold water pollution and whatnot. And now it's got these beautiful, great expanses of quality in-stream habitat, both in-stream and also riparian. And this is off the back of a lot of previous investment in river health programs in terms of riparian restoration and re-snagging. We've also got a highly engaged community. This was off the back of some of the work that Fern Hames and Scott Raymond and the CMA led up there with a demonstration reach, where we engaged the community. They were engaged in the importance of healthy habitats for fish, and it's the only pathway forward, I guess. And finally, we wanted to learn from the success that's happened in the Ovens River from trout cod, another nationally endangered species that was lost in the Ovens River, but off the back of a 10-year stocking program, we managed to get a self-sustaining population back into this system. And this program also included some of those genetic considerations that Sasha was talking about before. So we did it for trout cod, it's been successful. So let's see if we can repeat it with Macquarie perch. So what did we do? So in 2014, we kicked off a 5-year program where we wanted to stock and translocate fish. So over 94,000 fingerlings were produced over this 5-year period at the captive breeding facility out at Snobs Creek. So they were released into the ovens. We released over 2,200 largely juvenile fish that were translocated from Lake Dartmouth. And there was also brood fish that were used for the production of those fingerlings in the hatchery were also released there. Every one of those fish that was translocated and the adults that we used to produce those fingerlings all had a fin clip and their genetics, a genetic sample taken. So the source of these fish that we use, either as brood fish or for translocations, were from 2 different populations. One's Lake Dartmouth, which is our largest endemic population left in the Murray-Darling Basin. Their genetics is probably the 2nd or 3rd best that we've got left out there, but there's a lot of fish in this system, so we could, hence why we use this as our main source of brood fish and also fish for translocations. The other was the Yarra River, which is actually a translocated population outside its natural range, that was founded back in the 1920s and 1930s by the release of hundreds or tens of thousands of fish that were from the Goulburn Broken catchment. And fortunately, the fish in the Yarra River maintain all of that historical genetic material that's largely been lost back in the Goulburn Broken catchment. So they're a really valuable little population. Even though it's a translocated population, they're of immense genetic value. So these fish, both the fingerlings and also those that were translocated, were released across around 66 kilometres of river between Wangaratta and Myrtleford. And as I mentioned, we've released these fish from fingerlings, which are about 3 months old, right up to those ex-brood fish that could be 15 to 20 years old. So in terms of those individuals, for those stocked fingerlings, as I mentioned, they were the product of fish that were bred that were from Lake Dartmouth, and most of the translocated fish were all from Lake Dartmouth. There was a handful of those ex-brood fish from the Yarra River that were also released into the Ovens River. But interestingly and importantly, we also, and when I say we, the guys out at Victorian Fisheries Authority, also crossed the individual brood fish from Lake Dartmouth with the Yarra fish, both males and female crosses, to that genetic admixture that Sasha was talking about earlier. So it was set up what we were hoping to really bump up that genetic health of this, hopefully, population that we're aiming to establish. Of course, us researchers being lazy buggers, we didn't release all these fish ourselves. We enlisted the help of all the community groups out there, which was fantastic. So we had school groups, local land care groups, landholders all out there releasing fish in some of these great spots in the Mid-Ovens River. Further to this, and this was off the back of some of the great work that Glenn Johnson from Delft up in the Hume region and his team up there were leading, did an amazing job with the community engagement, touching base with all the key groups up there, media releases. They gave talks, set up barbecues to school groups, put up signage right across the release points throughout the Ovens River, which has been a huge part of this project. So what happened after 5 years? Are we on track to achieving our main objective, which is basically to re-establish a self-sustaining population of Macquarie perch? We wanted to do this by fish surveys and an assessment of the genetics of the population. And we were fortunate enough, and unlike a lot of programs, to have a really strong evaluation component of this program. So we've been out there electrofishing the river since 2016 as part of a few collaborative projects, as well as a full-blown assessment of 32 sites across nearly 200 kilometres of the Ovens and some of its tributaries, surveying to see were the fish that we were releasing, have they survived, and who were they? As I mentioned before, just like the fish that we used for brood fish, just like those that we use for translocations, any Macquarie perch that we come across in the Ovens River were also measured, weighed, and also we got a small fin tissue sample so we could work out some key questions. As I mentioned, we then compared the results of that genetics with those samples taken from all the released fish or their parents. So that leads to the specific genetic assessment. So by using all the genetic samples taken from all those fish that were released and any that were caught in the Ovens, they were all genotyped using diversity array technology. And that included the broodstock that were produced fingerlings, a sample of the fingerlings that produced, that's essentially our control, all the translocated individuals and the Ovens fish. We then used parentage analysis and identity analysis to identify whether fish that we caught in the Ovens River were stocked, were they translocated, or were they natural recruits in there. If they were stocked, we could tell what year they were released, therefore their size and age and subsequent survival and growth. And also, if they were natural recruits, we could work out who the successful parents were. So this sort of set up some really neat hypotheses that we had. So basically, and off the back of some of that work that Sasha just produced, were those individuals that were more genetically diverse, did they have better fitness? So we expected higher fitness of the Yarra fish and those that were crosses of Yarra and Dartmouth fish due to their high genetic diversity. And from what population did we expect? And as I just mentioned, we expected this to be in that Yarra fish or their crosses with the Dartmouth fish to be much better, have higher fitness due to them coming from a riverine environment, unlike the Dartmouth fish that come from a lake environment, given we were releasing them into a river, the Ovens River. So what did we find? Well, firstly, it was great. We caught Macquarie perch. So in the last year of the evaluation, we caught nearly 170 individuals in 2018, which is great. We weren't just popping them in the river and feeding the local trout cod and Murray cod population. What was great is that we detected Macquarie perch across more than 100 kilometres of river. We detected them at every one of our release points. We detected them at all the sites in between our release points, so they were dispersing. And what was awesome was that we detected them up to over 20 kilometres upstream of their closest release point, both in the Ovens River, but we also found them pushing 20 odd kilometres up the Buffalo River. It was great. And just to fast forward into what some observations we've had the last year or two, we've actually detected them up the King River, like tens or it's probably over 50 kilometres up the King River up near Edi, which is amazing. So those fish that we detected were, as we were hoping, covered a really broad size structure. They were from several months old right up to large adults, nearly 40 centimetres. So that was part of it. One of our objectives is to set up a broad size and subsequent age structure, just like the populations, those natural populations out there that remain. But then this comes to the interesting part. What did our genetics assessment show? What was amazing is that of all the fish that we caught, nearly 70% of these fish were locally born. So we had evidence that after just 5 years, we had evidence of natural recruitment that was occurring already. And you look at the just 1% of the total fish caught that were assigned as translocated fish. These guys were super important in those natural recruits, as I'll mention in a minute. So of those fish that were stocked, who were they? So basically we could see that the majority of fish that were stocked, and after we considered age-based survival and whatnot, the highest survival of offspring belonged to those fish that were either Yarra or the Yarra-Dartmouth crosses. And this is despite those fish only making up a much smaller proportion of the total fish that were stocked as well. So it basically gave us evidence that ancestry predicted survival and growth after stocking. So that's a couple of measures of fitness right there. Secondly, when we look at those fish that were locally born, our natural recruits that got us all so excited, most locally born fish had Yarra ancestry, then followed by mixed, then Dartmouth fish. So while those that were fish that were translocated into the river, and most of these, to be honest, were ex-brood fish. So only a handful of those fish were released in the river. They'd started breeding again within the river naturally. So ancestry also influenced natural recruitment success, which was fantastic. So if we flip back to our broad hypotheses, who were fitter? We expected higher fitness of Yarra and mixed offspring, and we also expected those fish from the Yarra to have much higher fitness due to them coming from those riverine environments similar to the Ovens. So we had both of those hypotheses were supported by our data. So the reasons for this are still quite speculative at the moment, but we assume that the Yarra fish are better adapted to harsher river conditions, if you think about the Yarra River, compared to Dartmouth that have potentially accumulated harmful variants under more relaxed selection, or they've lost some of their riverine adaptation, or they've started to adapt to lake environments. I guess what's clear though, and we just had a paper published recently, this led by the guys at Monash, was that using multiple sources really did show great benefits to both survival and recruitment of fish during these population reintroduction. So in conclusion, we were able to demonstrate that a population outcome for a threatened freshwater fish. And that genetic management component of this program was a really strong contributor to the success. We're able to help, and using this information, these learnings, we're able to help refine future actions, not only for Macquarie perch, but for other species moving forward. So there's a lot of discussions underway in that space. But of course, this is only early days. We're only 5 years in. We're still continuing those actions and monitoring. But I guess more broadly, it is a real blueprint to expand these actions out elsewhere. Sort of highlighted the importance of focusing on large systems. It's got to be a sustained effort, just like those learnings from trout cod in the Ovens River several decades ago. Importantly, we need to incorporate these genetic considerations where recommended. And this program was great in that we had this strong scientific foundation that included that evaluation component. As I mentioned, the work that Glenn and his team did, the community support and education was fantastic and was such an important part of the program. But we can do this, we can move forward and do this continue to do this for Macquarie perch, but also other species, but it needs support. This project wasn't a hugely expensive project, but the results were incredible. So we're really pushing to continue this momentum. And thanks everyone. And finally, I just want to acknowledge all those individuals and also stakeholder groups that I mentioned earlier and those that are on this slide. It was a fantastic effort from everyone involved.
Fantastic. Thank you so much, Zeb. That was fantastic. Really interesting to see the mix between the the genetics, the standard management, tying it all together, the community engagement, obviously a hugely important component of this. Just so impressive. How many years do you think that you'd need to keep on monitoring this system to make sure that those gains that you've got are going to sort of, be sustained over the longer term?
I guess there's two parts to that, Lindy. There's the monitoring, and we're fortunate enough to have a good long-term monitoring program already in the system, not just for Macquarie perch, but for the broader fish community as a whole, given all the work that has and continues to go on in the system. So we've got some of that monitoring happening already, but some of the actions are still continuing. There's still been stocking going on the last few years since this data was analysed, I guess, because the trout cod program was a 10-year program, and now no actions in terms of stocking has occurred for the last 2 decades because we've got this self-sustaining population of trout cod. Macquarie perch are no different. They're a long-lived species, 30-year-old fish. It's not like we can just drop a few in and walk away and forget about it. So we're continuing some of this stocking, but hopefully, fingers crossed, we can see some of that soon and shift to other systems where we're planning that right now. So, but yeah, we'll still be monitoring in the background for sure.
Fantastic. Thank you so much. Okay, I'll hand over to Andy now with some questions.
Wonderful. I'll wait for the video to catch up before you see my face, but I've got one question here for you, Zeb, about the importance of snags and in-stream habitat to the persistence in the river system. So you mentioned in your talk that the riparian veg and the new woody habitat was really important. Has it been quantified how important that was into this project?
I wouldn't say it's been quantified exactly for this project. We just published an article, I think it was 18 months ago, where we actually did quantify the correlated things like in-stream woody habitat density with the density of fish in a particular region. We got some really neat, significant associations there, and so we can actually predict now how much of in terms of carrying capacity or fish abundance or biomass within a reach can be attributed to the habitat load in the system. So we do actually have those quantified links, not just for the ovens, but for a number of systems. And we're actually incorporating some of those quantified links into some population modelling that John Yen's leading, where we're actually using that to prioritise what systems, where we go next for this kind of thing. So yeah, it is being thought about, it is being considered for sure.
Awesome, thank you. Relating to the fish that you're monitoring, what was the measurement that you used for fitness in fish?
So for the genetics, that was where we use growth and survival. So for survival, we use the measurement where we look at year class strength, and that's calculated. Let's say we dropped 10,000 fish into the system 5 years ago. We assign the average, because obviously if you drop 10,000 fish in 5 years ago, once fish hit 5 years old, there's generally not 10,000 left. There's natural mortality that goes on, but we look at that variation in mortality after however old the year class is that we're looking at, and that's one measure of fitness. So survival. Another one's growth, as I mentioned before. So we compared the growth rates of those fish that were mixed Yarra or Dartmouth, and so we compared the growth rates, so the time they were released with the size of the fish right now. So they're the two main ones we use for this study, but there's a whole lot of other measures of fitness that you can use, but they were just the ones that we chose for this program.
Very well answered, Zeb. Thank you. I've got a question here around the types of fish that you can use this sort of approach for. The question is around macas being quite an attractive game fish. Is there an opportunity to maybe extend this to less attractive fish, or fish that aren't used as a game or recreational species, such as galaxids? Absolutely, and a lot of that thinking's happened, is happening right now, and you may have heard about a conservation hatchery setup that's happening in Snobs Creek at the moment that's going to incorporate all those threatened, or a number of those threatened galaxid species, pygmy perch species and things. So absolutely, great question, and yes, it is being considered and hopefully acted. We see some outcomes from that in 5 years' time. We'll see. Perfect. I'm excited for it. I'm sure everyone else here is too. Last question. This is a bit more about the ecology of macas. This person's asking, are they generally associated with cooler waters and low salinity systems, or are they sort of a bit more adaptable than that? I would say yes, low salinity systems, but I guess when everyone realised, it was noticed the huge declines in abundance and distribution of macas, it was sort of, right on the cusp of, or right on the tipping point, I guess, and where those species, where macas were left was in a lot of our unregulated tributaries. Like we think about the Goulburn River tributaries, so the King Parrot Creek, the Upper Yea River, the Upper Hughes Creek. And so it was kind of thought that, it's those upper small systems that was their stronghold. And so that's where a lot of the early action sort of focused in on that, when actually historically the huge populations that have been documented were more associated with the main tributaries, the bigger river reaches, that unfortunately most of those sort of strongholds lie smack in the middle of where all our large dams are right now. So by focusing in on initially those small unregulated tributaries, it really sets us up for failure, I guess, in terms of climate change or episodic event like a bushfire and things, whereas actually those kind of events used to happen historically, but if you had a great big giant population back in the main stem of the Murray River or the mid Goulburn River, whatever it might be, there's your source for recovery, or to address those sort of those events. So that's the reason why we're actually now thinking more about those larger systems again, or those larger river reaches, to try and get them going in there. It's going to take more investment, it's going to take more fish to get them going, but it'll be a much more resilient population for the species moving forward, I guess. Thank you so much, Zeb. That was a massive answer to the question, and I really appreciate the detail you went to in that. I'm going to throw it back to Lindy to just say goodbye to you all. Lindy, I'm throwing it to you.
Okay, so thank you both Zeb and Sasha for fantastic presentations. They were both really interesting and shows just how fantastic these sorts of techniques can be in terms of conservation management. And thanks everybody for joining in, and we'll see you at the next seminar. Bye for now.